Hi, I'm Dr. Manjari Bhalkar. I'm going to tell you about the structure of hemoglobin. So let us discuss structure of hemoglobin. It is a globular oligomeric protein containing heme and globin. So we are going to see the structure of heme first, followed by the structure of globin. All of you must be aware that normal range for hemoglobin in male is about 14 to 16 grams per 100 ml of blood, and in female it is little less, which is 13 to 15 grams in 100 ml of blood. So let us see this very important globular oligomeric protein present in our blood in such a high concentration. It is essential to know its structure. So oligomeric means basically it has more than one monomers present. Okay, so there are four monomers, four globin chains that come together to form a hemoglobin structure. So before we move to that quaternary type of oligomeric structure, we need to know what is heme first, and then we will see what is how it is associated with globin structure. So when a question comes to you that what is heme? So chemically or biochemically, heme is an ion containing protoporphyrin 9. So now the question comes, what is protoporphyrin 9? So protoporphyrin 9 is made up of four pyrrole rings that are joined by a methynyl bridge. Methynyl means you have a CH group, and this carbon can get attached on one side by having a single bond, and on another side, another atom, with the help of a double bond. So this is how the carbon will have three valencies satisfied, two on one side and one on the other side. And the fourth valency is satisfied by hydrogen. So this is a methynyl bridge. Now, in addition to this structure, that is four pyrrole rings attached to each other with methynyl, you also have some groups that are substituted on this pyrrole ring. And these groups are methyl group, vinyl group, and propionyl group substituted on the pyrrole ring. That makes protoporphyrin 9. And this protoporphyrin 9 plus heme or plus iron will make it a heme. Now, iron is always in a reduced state, that is plus 2 state, and you get formation of a heme. So it can be represented diagrammatically like this. This is a pyrrole ring. You can see it's a 5-membered ring with two double bonds and one nitrogen present amongst these 5 members. Okay. So this four such pyrrole rings are connected to each other by methynyl bridge. It will form protoporphyrin 9 when they are substituted with methyl vinyl and propionyl. When it takes ion at the center of this four pyrrole ring, it makes heme. Let us see the complete structure, and so that it is easier for you to imagine or understand. So look at this structure. You have this number 1, 2, 3, 4. These are the four pyrrole rings. You can see that the pyrrole nitrogen is attached to the ion which is in the reduced state at the center. Now these four pyrrole rings, you can see that they are linked to each other by methynyl bridge. This asterisk mark indicates presence of CH. And so at one side, at one ring, it is attached by a single bond, and to the other ring, it is attached with the help of a double bond. Now look at the substitutions over here. The first pyrrole ring is substituted with methyl group and vinyl group. CH double bond CH2 is a vinyl group. So let us see these substitutions in a clockwise fashion. So ring 1 is methyl vinyl substituted. Ring 2 is also methyl vinyl substituted. Ring 3 is methyl propionyl substituted. And ring 4 is propionyl methyl substituted. Is this clear? So this is a protoporphyrin 9 plus iron that makes a structure of heme. Now this particular structure, we can write it in a simpler format this way. And this is called as a Fisher shorthand formula. So you can, it is just represented by 1, 2, 3, 4, 4 pyrrole ring and iron at the center. And each of the pyrrole ring is substituted by certain groups. In a clockwise manner, you have methyl vinyl, methyl vinyl, methyl propionyl, and propionyl methyl. So while understanding these substitution, you will also understand that these substitutions are not in a symmetric fashion. Okay, so when the substitutions are in an asymmetric fashion, it is called as type 3 uh, porphyrin. Okay, so this is a type 3 porphyrin with asymmetric substitution. Now you also have certain porphyrins present in the nature which are type 1 porphyrin. That means these various substitutions are in a symmetrical fashion. Okay. So in the nature, we get either type 1 that is symmetric substitution or type 3 that is asymmetric substitution of the porphyrins. So four pyrrole group together, it is called as a porphyrin ring structure. Now there are examples of porphyrins. You can have uroporphyrin 1 and 3. So uroporphyrin symmetrical and asymmetrical, where you have substitutions like acetyl group and propionyl group. You can also have coproporphyrins 1 and 3, symmetric and asymmetric substitution of methyl group and propionyl group on the porphyrin ring. Is that clear? Now see, uroporphyrins and coproporphyrins are actually the intermediate structures generated at the time of heme biosynthesis in our body. Okay, so later on these intermediates finally they are converted to protoporphyrin, and protoporphyrin plus heme will generate a structure of heme. Now let us move further, that iron is linked to the 4 nitrogen of the protoporphyrin ring by coordinate bond. But iron can form 6 different coordinate bonds. Out of that, 4 coordinate bond is with 4 different nitrogens of the pyrrole ring. Now, fifth coordinate bond is formed between the nitrogen of the proximal histidine residue, that is F887 of globine polypeptide chain. Now, I want you to stretch your imagination further, that this heme is attached to a globine chain in a hemoglobin structure. Okay, so imagine this as your globine chain made up of several amino acids. Okay, so each of this globine chain is folded in such a way that you get formation of a pocket. Now, in this particular pocket, there are all hydrophobic amino acids present. And in this pocket of globine chain made up of amino acids, you have a heme which is situated inside the pocket. So imagine this as a heme. Okay, so this has 4 pyrrole rings at the 4 ends and iron at the center. So this heme, when it is situated inside the globine pocket, now you can imagine that iron at the center has formed 4 coordinate bonds with the 4-pyrrole nitrogen. Now, this ion can form the 5th coordinate bond with the histidine amino acid of the globine chain. Okay, and this histidine amino acid is the 87 number amino acid of the globine chain. And this is forming a coordinate bond with the ion of the heme. So that is how these heme and globine chains are linked together. Is this clear? Right. So this is how the 5th coordinate bond is formed. Now what about the 6th coordinate bond of the ion? This 6th coordinate bond can be formed with oxygen or water or carbon monoxide. That is how the hemoglobin can carry the oxygen. Okay, so oxygen can be attached to this reduced state of iron, and it can donate the oxygen and becomes deoxygenated. So hemoglobin can be oxygenated and it can be deoxygenated on delivering the oxygen to the tissues. So you will have oxygenated hemoglobin in the lungs and deoxygenated hemoglobin generated in the tissues on donating the oxygen. But remember, the site for binding of oxygen to the hemoglobin is always to the ion of heme. Okay, and it is the same site where carbon monoxide also binds, and that is where the danger lies. The carbon monoxide has higher affinity with the ion of the heme compared to the oxygen. So carbon monoxide can just displace the oxygen and form carbon monoxide linked hemoglobin, and then it cannot transport the oxygen, and which is a condition called as a clinical condition which needs a hyperbaric oxygen treatment. Okay, that we will discuss later on. For now, you can remember that here in this, again, let us imagine the same structure. This is a globine chain folded, forming a hydrophobic cleft, and in that cleft you have the heme ring situated. The heme ring is attached on one side to the histidine number 87, and the ion is also linked to oxygen over here. Now further, this oxygen is also linked to the globine chain with the help of another histidine which is located on the globine chain at number 58. And this 58 number uh, histidine is called as a distal histidine. So understand that 87 number histidine is a proximal histidine which is in close proximity to the heme, close proximity to the iron. And 58 number histidine is a distal histidine which is not in very close proximity. And you have oxygen in between. Okay, so iron, then oxygen, and then you have the distal histidine located. This distal histidine and proximal histidine, we are also calling them as F887, which is a proximal histidine, and E758, which is a distal histidine. So you must be wondering, what is this F8 or E7? So I want to tell you that when we are discussing about the structure of this globine chain, this globine chain is folded to have a tertiary structure, but here you have a secondary structure in the form of alpha helices. Now, each of these alpha helices, they are given alphabetical name like A, B, C, D. So actually, this 87 number histidine is located on the alpha helix F, and in that alpha helix F, It is the eighth amino acid. Okay, so this is called as F8 but 87. Similarly, the distal histidine, its number is 58 in the entire globin chain, but it is located on the E alpha helices, alpha helix E, and it is number 7 on the E alpha helix. Now, F8 and E7 histidine, they lie on the opposite side of the heme ring. So again, when you are imagining this, you can always notice that you have a heme situated, a heme ring or porphyrin ring situated into the globin chain pocket. And on the one side, you have the proximal histidine, and on the other side you have the distal histidine. So they are located on the opposite side of the heme ring. So now little more detail about the globin chain. Now globin chains, they are present in most of the form of the hemoglobin are two alpha chains. So we always have one hemoglobin made up of two alpha chains, and then it is also made up of two other chains, that is it can be beta, gamma, delta, epsilon or zeta chain. For example, in adult hemoglobin, you have the 98% of the adult hemoglobin contains two alpha and two beta chain. Okay. Now each of this alpha chain is made up of 141 amino acids, and that indicates the primary structure of a protein. So understanding the structure of a globin chain, which is a protein structure, so you have to know that primary protein structure indicates the number and the sequence of amino acid present in it. So alpha globin chain contains 141 amino acids, and they are arranged in a secondary structure which is made up of alpha helices, alpha helix structure. The alpha helix structure is always stabilized with the help of hydrogen bonding between first amino acid and the fourth amino acid. That is how it will form a spring-like or helix kind of a structure. So in each of this globin chain, in each of this alpha chain, you have 7 alpha helices. And each of these alpha helices, they are given a specific alphabet from A to H except D. Okay. So that makes 7 alpha helices, and that will indicate the secondary structure of a globin chain. Now, when it comes to the other two chains in one hemoglobin, other two chains can be beta, gamma, delta, epsilon, or zeta. Now, these chains, they have 146 amino acids, and there are 8 alpha helices present. And here you will have the D alpha helices also. So, they are also labeled with alphabets A to H. Now, one heme is attached to each polypeptide chain. That means in one hemoglobin structure, you have 4 heme and 4 globin chain. Is that clear? Now, each of the heme is also made up of 4 pyrrole ring. So, it is a 4-4-4 type of a structure. So, 4 pyrrole ring, 1 porphyrin. 4 porphyrin, that is 4 heme, and then you have 4 globin chains. So, that completes the structure of hemoglobin. Now, globin chain is then further folded, that is tertiary structure, and it is folded in such a way that it creates a hydrophobic pocket where you have inside the pocket, all the amino acids or most of the amino acids are non-polar, which makes this pocket hydrophobic. And why functionally this is important? Because then it can protect the iron from getting oxidized. So suppose if this iron gets oxidized to Fe plus 3 state, then it cannot bind with the oxygen and it cannot carry the oxygen. So such a hemoglobin is called as a met hemoglobin or oxidized hemoglobin. So in order to avoid that type of a hemoglobin formation, there is a always hydrophobic pocket which is generated by the hydrophobic or non-polar amino acids of the globin chain. Now this pocket will also permit the reversible binding of oxygen to the iron. Now, four globin chains with four heme located together, they are characterized by quaternary structure. So this will stabilize the quaternary structure by non-covalent bonds like van der Waals forces and hydrophobic interactions. So what are the interactions between these four different globin chains to keep them together to make a functional unit of hemoglobin? So these are the forces that require to keep these four different globin chains together, and these four different globin chains will contain four different heme inside them. Okay. So let us see this type of a structure of hemoglobin described. You can see the diagrammatical representation. There are two red globin chains. If you decide that they are the alpha globin chains, they will have 141 amino acid with, you can see this spring-like helical structure. So A to H except D. Now green one are the beta globin chain, or they can be delta or epsilon or zeta type of a globin chain structure with 146 amino acid, and there are 8 alpha helices which are present in the other type of globin chain. Now inside the hydrophobic pocket of each of this globin chain, there is a heme which is situated. Okay. Now you can see that there is an interaction between the adjacent globin chains, and these interactions are non-covalent. That keeps them together as a quaternary structure. Now each of the alpha globin chain will interact with the other type of or beta chain, and there is no direct interaction between the two alpha globin chain or two beta globin chain. Okay, so the interactions will be only between the alpha and the beta, etc. There is no interaction between beta beta or alpha alpha type of a similar type of a globin chain. Now you can also notice that there is a hollow pocket at the center which enables the exchange of gases with the hemoglobin. So there is no direct contact point between the two alpha or two beta chain, but there are several contact points between the alpha beta chains of the dissimilar chain pairs. And there is a central open channel or cavity in the hemoglobin molecule. So I hope that you have understood the structure of hemoglobin. In the next video, we will discuss the various normal types of hemoglobins, and we will also discuss the formation of oxyhemoglobin, formation, the dissociation of oxyhemoglobin, etc., and the functions of hemoglobin. Thank you.